It's a really huge pleasure to be able to introduce Chris Somerville tonight as the first of 3 keynote speakers. Always hard to get keynote speakers, but last year's keynote is now Secretary of Energy. It was easier this year. Chris's career is hard to summarize in 30 seconds, but I'll do the best I can. Chris got his initial training at the University of Alberta, did some time at Michigan State University before landing at the Carnegie Institute at Stanford, where he's been their director since the mid-90s. He's made two numerous seminal contributions to our understanding of plant biology, including our understanding of plant wall synthesis, to any real justice to. But he also was early to the table in understanding the power of plant science to be translated into sustainable crops to feed the planet. And through the foundation of Mendel Biotechnology, which had sustainability and that translation component of its founding piece, they showed really the path forward. Chris was early to recognize, Chris and his company were quick to recognize that the corn-based ethanol was going to cause land use problems, and the path forward for really replacing biofuels was with cellulosic substrates rather than corn, and that is now a part of Mendel, as well as Chris's own work in basic science realm. Steve Chu pulled off a major coup in getting Chris to move to the Energy Biosciences Institute at LBL and UC Berkeley a couple of years ago, where he now serves as EBI's director. You'll notice the Somerville thumbprint all over DOE's roadmap for cellulosic biofuels. Chris had a major hand in writing of the biofuels roadmap for DOE. And he's going to talk to us tonight about the development of cellulosic biofuels. Chris?
Thanks a lot. We had a very interesting experience at Berkeley when we founded the EBI. Although there were a group of us that had some interest in energy, we thought we should ask our colleagues to make sure we had found everyone who might be interested. So we posted a document that I've been involved in writing called Breaking the Barriers to Cellulosic Ethanol on our website. And we got and we invited proposals from all the faculty at LBL, Berkeley, and University of Illinois. And we got 250 proposals from about 700 faculty members back, with a really wide range of ideas. And of course, we went through a process to narrow that down to the roughly 50 groups that we now support through the EBI. But one of the things I learned from that experience was that it can be very productive to educate people who don't know a topic about what the problems are. And what I mean by that is that, for example, EBI now has a large chemistry activity. We have about half a dozen really leading chemists working on problems in bioenergy, none of whom had worked on it before we invited their participation. And I now recognize that, in fact, well, I think we probably all recognize how difficult it is to move into a new field if you don't know what the core problems are and what the issues are. So what I thought I'd do tonight is just give you kind of a very quick overview of some of the issues that seem most relevant to this group. And I've included a few things that they're not directly, not even indirectly related to genomics, but you need to understand in order to see the big picture, I think. So I'm going to begin here. This is the options for decarbonizing the energy supply, I guess is the way to say it. And to give you a sense of what the problem is, I've put nuclear over on one side, and the reason is that energy use is expanding by about 1 gigawatt, 1 billion watts every 2 days. It turns out 1 billion watts is the size of the average nuclear power plant. So if we wanted to keep CO2 emissions where they are by going nuclear, we would need to build one every 2 days. And that gives you a sense of what the fundamental problem is. Of course, we need to decrease emissions, not just stay where we are. And according to DOE, the other options are shown here. We use about almost 14 terawatts. That's the instantaneous energy use rate. So a terawatt, of course, is 10 to the 12. And if you look at what the options are, we pretty much used the hydro. We could get 2 terawatts of tides and currents. DOE thinks 4 terawatts of wind, 12 terawatts of geothermal. But of course, the big opportunity is shown here. That is, we get about 10,000 times as much energy from the sun each year strikes the surface of the planet as all human energy use. So if you really get down to it, the only option to really decarbonize on a large scale, barring geological sequestration, is solar. Although all these other options are also good and need to be approached in parallel. In particular, wind and geothermal are pretty mature and are excellent technologies, so I don't need to say there's only one solution. Now, I'm not going to talk about photovoltaics. As you probably know, photovoltaics have higher efficiency, and I hope that the long-term solution will be either photovoltaics or some sort of photoelectric material. But to meet U.S. energy needs, 3.3 terawatts, we need about 26,000 square kilometers of photovoltaic devices, the best current devices. But in the history of the planet, the total manufacturing amount is 4 square kilometers. So we're thousands of fold away from where we need to be. And although, as I said, I hope we'll break that barrier sometime, it's not imminent. Really, the other option for harvesting photons is shown here. That is, plants use the energy in sunlight to reduce carbon dioxide to polysaccharides, and when they're burned directly or indirectly, the heat release can be used for work. And in theory, the CO2 cycles around. So in theory, it's carbon neutral. In reality, it's a lot more complicated than that. If just tilling the soil by introducing oxygen leads to microbial activity and off-gassing of both CO2 and nitrous oxide. If any land conversion is associated with that, it can be extremely negative for CO2. That is, if you cut down trees and burn them in the course of clearing land. Fertilizer use and production is very negative, so about 2% of energy is used to make nitrogen fertilizer worldwide, and about up to 3% of that off gases as nitrous oxide, which has a CO2 or a greenhouse gas footprint 250 times bigger than CO2. And of course, then there's all the transportation and processing associated with turning biomass into fuels, and so it's not every way of using plants to harvest sunlight is actually meets our goals in terms of producing carbon neutral or even low carbon fuels. Now, just to give you a sense of what this is all about, this slide is the result of a meta-analysis of 537 studies where land use change, the impact of land use change on soil carbon has been measured. And it's not all bad news. In fact, what this shows is pretty much you can increase soil carbon by planting perennial grasses or in some cases trees. It shows that perennial grasses sequester more carbon than trees, and if you plow the soil, you go negative. So those are the basic rules from a very large number of studies, and I'll come back to that in a moment. In addition, however, recently it's become apparent that there is another component to this called the indirect land use. So what I just showed you might be part of the direct land use, but the most bothersome part of the current discussion about land use is the indirect effects shown here. And it's a fairly simple concept put forth by Tim Searchinger last year in a science paper, and the idea is that if you take an acre of corn and divert it to producing fuel, because the demand for food is inelastic, someone somewhere else in the world will try to produce that corn. And with some probability, they'll take an acre of undeveloped land to produce, or part of an acre to produce that corn. And when they take some piece of undeveloped land, they'll cut the trees down and burn them, and then they'll till the soil and off-gas the carbon stored in the soil. And so, to the extent to which this actually happens, this negates the value, seriously negates the value of biofuels, any type, for, in some cases, hundreds of years, depending on the exact application. So we sponsored actually 9 workshops on this topic last year because it's an important and difficult concept. And what makes it difficult is that it's an entirely economic argument. based on what's called the general equilibrium model. And general equilibrium models depend on having realistic assessments for what are called market-mediated effects. That is, how does a market respond to a market signal? And I don't really have time, and I don't want to give a whole talk on this subject, but just in case you hear about it, it's the reason it generates so much discussion is that you may be aware of the fact that economists don't know up from down, as far as I can tell. Our current economy would be a kind of market signal of itself, indicating that economists actually don't have a fix exactly on what's really going on in the world. And so that's what partially generates the discussion. There's a germ of truth in it, however, and I think that what it really reflects is that, of course, we don't want to cut down forests to make fuels. We don't want to take natural ecosystems that have a lot of carbon stored on them and convert them to fuels. The big question is that what's going to happen if we direct land towards it that's not already used for it? And I think the flaw, and the fundamental flaw in the argument is that because we don't value natural ecosystems, it's very hard to use market-mediated effects to stop them from being developed. And actually, where this is really going to lead, I think, is to some sort of an international agreement that will finally protect natural ecosystems so that market-mediated effects can then take place on the other acres. And I think that's where this has to go in the long run. Now, concerns about the land use and also the related food for fuel debates, of course, have stimulated lots of interest in algae, and I guess you had a talk here today that I didn't get here in time for. We're not actually pursuing this to a significant extent in EBI, but I do want to point out that it's not exactly that algae, although they can be more productive than plants at theoretical production, they're also pretty big users of land and water, and so it remains to be seen whether this is actually going to be a good idea. There's currently a moratorium on the use of federal desert land for solar thermal production, because many people don't see desert land as wasteland. So I think there's going to be challenges using algae as well. In this country, the government has, in 2007 at least, the government introduced an act that pretty much determines now how developers of biofuels get subsidized for that activity. And this is a manifestation of it. What it's showing is that corn farmers will get subsidized at 50 cents a gallon up to the production of about 15 billion gallons. You wonder, well, why 15 billion? How did they come up with that number? And the answer is that the fleet, the light vehicle fleet in the United States will use about 10% ethanol in the fuel. And that fleet uses about 140 billion gallons, so that would be 14 billion, and they threw in $1 billion for good measure. So the corn farmers think they can actually produce all the ethanol required to sort of saturate the fleet. Now, the Act also has a subsidy for what are called advanced fuels, and these are biodiesel and algal fuels and cellulosics. And as you can see, the agenda is pretty aggressive, and that is the expectation is that this should reach whatever that is, 35 billion gallons by 2022. So, and I think the longer-term goal, the federal goal, would be to get about 30% of our transportation fuel from biomass by 2030. 20% would be about 65 billion gallons. So, to give you some sense of what that might mean, a fairly large bioconversion facility would produce 100 million gallons a year and would use a million tons of biomass. So to make 65 billion gallons, we're looking at 650 very large processing facilities. And the cost of installing those would be more than $325 billion. So that's the kind of change that's currently being contemplated when envisioning biofuels on a large scale. It remains to be seen. It's a stretch goal, I would say. Now, as to where the biomass might come from, many people will be aware that DOE and USDA collaborated a few years ago on something that's colloquially called the billion-ton study, shown here. And what they did was they looked around the country, county by county, at land use, and they estimated what was growing and what could be grown and how much of it could be grown, and they came up with this pie chart. And so, for example, they estimated that if we took half of the corn stover from the 90 million acres of corn, we could produce about 26 billion gallons, assuming we got near theoretical efficiency. The problem with this is that although that's true, the biomass is there in large amounts. Corn farmers are really busy when they're harvesting corn. It's a narrow window. Generally, they call up their 80-year-old relatives from the retirement homes and get them to drive tractors for that couple of weeks, and labor is actually in pretty short supply. So if they're going to actually take the corn cobs and stover off in addition to the grain, they're going to need about 10 more relatives per section. In other words, there's a real problem, and they can't just throw it on the ground and pick it up later, as it turns out. That's technically hard to do. So I'm not so sure that this is a realistic possibility. Similarly, the wheat straw, there's only 1 ton per acre of wheat straw, roughly, and there's a very non-linear relationship between tons per acre shown here and the cost per ton, so shown here. So you can see down at 1 ton, 1 ton per acre, just the cost of transportation and collection would add about $1 a gallon to the cost of fuel. Whereas if you're out here at 15 tons, it's about 10 cents a gallon for collection and transport. So, I think we take this pie chart with a kind of a grain of salt. It does illustrate that there's quite a bit of biomass, 1.3 billion tons. If we actually could collect it and convert it to fuels, that would be about 130 billion gallons. So it's a substantial amount, but probably not as shown here. The biggest piece of this is this part here, and this reflects the fact that we pay farmers not to farm about 40 million acres in the United States. It's mostly in perennial grasses, and these authors assumed that with some work we could put more highly productive perennial grasses on those acres. Switchgrass was what they proposed, or something like it, at 8 tons per acre, and that would produce by itself about 45 billion gallons. Now that's probably the most reasonable proposal, I think, in this. I just want to point something out that's kind of interesting, and that is, according to indirect land use rationale, by not growing corn on those set-aside acres, we're actually deforesting the Amazon. That's the rationale, if you follow the logic through. And so that's one of the problems with the overall idea, I think. So, additionally, I should say that we don't have just an American view of this. If one takes a larger view, the big opportunity is clearly sugarcane as a cellulosic fuel rather than just as a sugar fuel. So at present, you probably know that the Brazilians produce 40% of their transportation fuels from sugarcane on less than 4 million hectares. And just to put that in perspective, in Brazil there's about 7 million hectares of sugarcane in total. About half of it's used for fuel. And by contrast, there's about 234 million hectares of cattle ranch. So the amount used for sugarcane is actually far away from the forest, mostly down near Sao Paulo, and it's a very small proportion of their agricultural acreage. My Brazilian colleagues that have been studying this claim that this region, which is mostly used for cattle ranching, could expand up to 80 million hectares of sugarcane. And if you used it as a cellulosic crop, our estimate is that would produce between 1/3 and half of all the transportation fuel in the world. So long term, no matter what we do here in the United States in terms of biofuels, really the large opportunity is actually in Brazil. And so sugarcane, if you care about what's the most important plant, it's unambiguously sugarcane, or a related variety called energy cane, a variety that doesn't produce much sugar. Now, in this country, there is a small region that can be used for sugarcane along the Gulf Coast, and indeed some of the first commercial plants, the one that BP is working on at present, is a sugarcane cellulosic plant. There are many species that can be used, and I think what we're going to see as if this unfolds in scale, is we're going to create a large number of new energy crops. You probably can't see this, as I recognize having sat in the back of the room, but what it's showing is simply a list from a paper in Biomass Bioenergy a couple of years ago of plants that have been studied to date. And some of them can be pretty highly productive. Here's energy cane. They're estimating 32 metric tons per hectare per year. Actually, that seems low to me, since sugarcane in Brazil yields about 120 tons per hectare. The only point I wanted to make is something like the following, that pretty much everything except for sugarcane that we're going to use as an energy crop is more or less undeveloped. And so there's many opportunities to apply the tools of genomics to more rapidly develop these as crops, and I'll mention a couple of things in a moment. My favorite among the many undeveloped species is shown here. This is Miscanthus Giganteus, a rather famous picture by now, I think. A normal-sized person standing in front of it, Emily Heaton. And this is a 8-year-old plot growing at the University of Illinois. Every year it's been harvested at ground level, all the biomass cut at ground level and taken off. It's never been fertilized, it's never been irrigated, and it's not sprayed with anything. And so environmentally, it's pretty attractive. And on an average year, you can drive around the world twice on an acre of this. That is, if you turn the biomass into fuel, liquid fuel, you could drive around the world twice in an average car. The best year got 26 tons, which at 100 gallons a ton, which is roughly the theoretical, or 90% of theoretical efficiency, that would be about 2,500 gallons. So that gives you a sense of what we're thinking of in terms of what an energy crop should look like and what it can do on a decent acre. It's not the most productive plant. There are others that have been measured that can be higher, like Napier grass. True elephant grass has been measured up to 45 dry tons per acre. And as I mentioned, sugarcane can go above 100. So this represents, just by the way, about 1.4% solar efficiency on an annualized basis. So even though it spends 3 months under the snow, if you integrate all the photons that end up in chemical energy, it's about 1.4%. Here's switchgrass, by the way, growing in front of it at Illinois. Switchgrass is about a third to a half as productive on the same acres. So we think wherever there's enough water, we'll be growing something like miscanthus, and on drier acres, probably switchgrass. One of the reasons that miscanthus has emerged recently as an interesting plant is the Europeans did a 10-year, 17-site study of it. And actually, in the context of using it to co-fire in power generating plants, something that is likely to continue. So we know quite a bit about it. You can see, or you may be able to see, all the sites throughout Europe where it was studied. And one of the nice things is it thrives, as you can see, all the way from Denmark to Greece and Portugal. One of the things that makes it attractive from environmental purposes is these and this plant and others like it are harvested late. So you can see there's snow on the ground here, and in this case it's being chipped, but it can be baled. And this is very attractive environmentally because this plant undergoes sort of programmed cell death and withdraws its mineral nutrients back into a big rhizome, so that actually when you're taking it off the field, you're taking 700 parts of carbon per 1 part of nitrogen. And so there's not a lot of nitrogen lost. And in fact, here's a 14-year study showing a lack of response to nitrogen over 14 years in England. So blue is no applied nitrogen, 60 pounds or 120 pounds. And over 14 years, they took all the biomass off every year. There was no response to applied nitrogen. We think there's two reasons for this. One is, as I mentioned, it's withdrawing mineral nutrients back into its rhizome. But in addition, we believe there's some sort of an endophyte that's capable of at least providing some nitrogen to the plant. In fact, this is the most exciting slide I saw last year, because the Brazilians have been claiming for many years that in some regions sugarcane doesn't need applied nitrogen either, and have identified microorganisms that are involved. And Angela Kent at University of Illinois has isolated an organism from miscanthus, and in fact has got a response on rice. And so we're kind of very excited and intrigued, and I think this has become for us a big priority, is to understand what's really going on here mechanistically. Because, yes, in the larger scheme of things, this is the kind of response that's exciting when it comes to improving, using biomass crops to actually improve agricultural practices. I spoke recently at the Chesapeake Bay Authority, where there's a large 4-state collective activity engaged in trying to clean up the Chesapeake Bay, which is heavily contaminated by mineral nutrient runoff from field crops. And they're now excited about the idea of actually planting these kinds of crops around the bay to actually capture mineral nutrients and prevent them from running into the bay. And I think there's going to be a lot of opportunities for that going forward. The priority research topics for Miscanthus and most of the other, if not all the other, perennial grasses that we care about are shown here. First thing is, since these are very long-lived, some of these perennial grasses stand in a field for up to 40 years. We really need to pay attention to pest and pathogen tolerance. And so I think we believe that unlike field crops, these things are going to be planted as multi-lines or as mixtures, genotypic mixtures, with a lot of genetic diversity in them. And so I was just telling Eddie that one of our big questions is, you know, we need, for each of these species, we need to get out there and measure diversity so that we can at least populate fields with known genetic diversity. Ultimately, we'd like to know the genetic basis for pest and pathogen resistance, and that will have to come later, I think. Additionally, these things are self-incompatible, so we really need to know the molecular basis for self-incompatibility so that we can generate parental materials so that right now these things are pretty much extremely heterogeneous. You couldn't breed them if you wanted to, basically, since you can't make homozygotes. And as I mentioned, we really would like to know what the mechanistic basis for the nitrogen response is, not only in Miscanthus, but in other members of the sugarcane family. Now, the way we see it, or at least the way I see it, I guess I should say, is that wherever the land is smooth enough, we're likely to use these grasses. And the reason is that, to my knowledge, there's no C4 tree. So grasses like Miscanthus and switchgrass are very water efficient compared to most other species. And since water is the limiting factor in most ecosystems, we think that the grasses will dominate. However, a lot of the land is, of course, too rough for machine harvesting, and so we believe trees will dominate there. We have 750 million acres, roughly, in the United States of trees, and you can see from these slides that not all of it is timber. So what this is showing is the blue bars are total trees, the green bars are timber, and these are the 4 regions of the country. You probably can't read this. On this slide, blue is public lands, probably not really available for any purpose other than recreation and ecology. But green is private, and what you can see is in the north and south of the United States, there's a tremendous amount of private woodlot, and so, and furthermore, a lot of that land is actually stranded. We've lost more than 200,000 jobs in the pulp and paper industry in the United States in the last 10 years due to rapidly growing eucalypts in South America, and so there's a lot of stranded wood available. It's frequently owned in fairly large lots as well, which facilitates its use for these kind of industrial purposes. So I think we will see quite a bit of development of some woody species. Some coppice and some perhaps longer rotation. Furthermore, of course, I already mentioned water is the limiting factor, so this is the rain map of the United States, and pretty much this is the 35-inch line right there. And 35 inches is enough to produce, let's call it 20 dry tons per year without irrigation. So this is the region that's most likely to be where all the biomass will be grown. Here it is worldwide, the same thing. In this case, blue is too cold, red is too hot. And so you can see actually Eastern Europe has the ability to produce more than 1 billion tons a year as well. China actually is quite a lot of land, although we're studying how much of that is truly available for access to food production, and of course South America and also Africa. There are large regions. So we believe that biomass will be produced around the world in fairly large amounts. Now, I think I've alluded already to the fact that there's competition between food and use of land, let's call it that. As I said, we don't want to convert any acres that are not already used for something, not already used for agriculture. And the question is, how much land does that open up if we're not going to cut any forests or savannas? And my colleague Chris Field and some of his students and postdocs recently published this paper, Environmental Science Technology, where they used global satellite images to ask that question. And their conclusion was that actually more than 1 billion acres worldwide have been used for agriculture and abandoned. So we actually feel moderately optimistic that there is a fair amount of land around. And here in the United States, for example, there's more than 100 million acres has been used for agriculture, and it's gone out of production mostly for economic reasons. As you know, or you may know, about 40% of farm income in the developed world is government subsidy. In fact, the big agricultural problem in the last 2 generations has been getting paid more than it costs to produce agricultural product. Perhaps we're transitioning out of that, but that did cause a lot of land to go out of production. 50 million acres, roughly 50 million acres since 1980. So we think there's a lot of land. Now, some of that land has actually gone out of production because it's been damaged. So here in California, for example, we have a large, more than 20,000 acres that's gone out of production because it became contaminated with selenium, which is incompatible with using it for food production. But it's actually quite compatible for using it for biomass production. Elsewhere in the world, there's a lot of arsenic-contaminated land and, of course, desalinized land. So what that means actually is that some of the species we're really interested in are species with good tolerance to harsh conditions, you might say. In that respect, Spartina, for example, the marsh grasses growing here around the bay are all Spartina species. But there are dry land species that exhibit comparable levels of salt tolerance. And some of them can be almost as productive as Miscanthus, cordgrass in the United States. Additionally, the other kinds of things that are of interest, of course, Jatropha is probably the most rapidly expanding crop species in the developing world because, actually this is Jatropha shown here. It produces an olaceous seed with a high oil content, but importantly, it's extremely drought tolerant. No plant can grow without water, but Jatropha can survive without water. And so it's being developed around the world as a biodiesel crop. Now, biodiesel is a really bad idea if you have good acres because every oil-producing plant has much lower productivity than a comparable, highly productive cellulosic plant. For example, in this country, soybean produces about 63 gallons per acre of oil. On that same acre, you can make 2,000 gallons of ethanol from Miscanthus. So if the land is good, you surely shouldn't be growing an oil crop. But if the land is challenging, actually Jatropha will be a good crop. And importantly, the technology for making cellulosic fuels is unlikely to reach the developing world for some generation, I would say a generation or two. It's a high technology, capital intensive. By contrast, you can make biodiesel in your bathtub with stuff you can buy at a local grocery store. And so for some regions of the world where energy is not readily available or is extremely expensive, such crops can be a good option. There are some additional organisms like Leucaena is a nitrogen-fixing tree, is extremely productive. That would be as a wood crop. Pongamaya is also a nitrogen fixer, grows on depleted soils, but actually produces a lot of oil, as does Copavina, or Copa Ifera. This is the one that Mel Calvin was waxing poetic about in the late 70s. So I just point this out because I think it's important that energy is a world problem, and I think we need to try and think about it in that context and get out of the mode that we've been in for the last couple of generations in the United States, I think, of largely being focused on American field crops and start looking at it as a world problem, because I think whatever happens in India and China and elsewhere in the world is just as important to us as what happens here when it comes to solving the climate problem. I want to switch gears now and just talk a little bit about some of the biotechnological aspects. I think the first thing I want to say, this is a very well-known slide that shows what you might call the thermochemical conversion of biomass to various fuels and other chemicals. And all I'm going to say about this is that all this technology was developed for coal and works quite well, is mature. This is how South Africa got fuels during the embargo. And it works today. Some of the commercial entities that are trying to make biofuels are using variants of this. So, for example, Range Fuels is actually using syngas to ethanol, and Coscada is using syngas to ethanol, but instead of using a catalyst, they're actually using an anaerobe, a microbial conversion. Otherwise, this technology is not readily, well, it has a problem, and that is it requires, it operates at high temperature and pressure, 1,000 degrees and 20 megapascals. And it turns out it's expensive to engineer that. And so in general, these facilities that carry out these kinds of reactions are built very big to take advantage of what's called the engineering rule, which is fundamentally that the bigger you build it, the cheaper per unit it gets, usually at the factor of 0.6 per unit. So it may well be that it can be scaled down, but in general, the field is turned towards bioconversion because bio scales much better, scales down much better anyway. You know, you can go flask to 200,000 liters relatively easily. And the steps in this process are probably well known, but in general, basically you grind up the biomass, and how fine you grind it matters. Then typically it's boiled in hot acid briefly, like 1% sulfuric acid for 2 minutes at 180 degrees. That causes some fragmentation of biomass, and the sugars released are then fermented in one step. The remaining solids are then usually treated with an enzyme, cellulases and xylanases, and the sugars released in that are fermented to produce, in this case, ethanol. And then remaining solids are usually burned, lignin and other solids are burned to make electricity. It's actually pretty complicated. I was tempted to show you the 21 process schematics for this, just to give you a sense of the fact that actually it looks easy, but the reason it costs $500 million to do 1 million gallons or 100 million gallons a year of it is that the engineering is actually quite complex. 21 different process components. Now, the reason, so this process has a lot of problems. Otherwise, we would have a lot more cellulosic ethanol kicking around. And one of the problems is that the hot acid treatment causes dehydration of sugars to materials, to compounds like hydroxymethylfurfural that are strongly inhibitory to growth of microorganisms. Secondly, a lot of the polysaccharides are acetylated, and that comes off as acetic acid, and that's also inhibitory to the growth of microorganisms. And this in turn means that in general, you can't arrive at that right balance of, fragmenting all the polysaccharides with acid, because you end up producing inhibitors the longer you hydrolyze it with acids. Secondly, the more of these inhibitors you put in, the less solids you can put in your fermenter, and therefore the more fermenters you have to have, because you're running them at low product yield, if you can understand what I mean. So in the field, the big problem is this pre-treatment step, finding a way to fragment the polysaccharides without creating inhibitory compounds, and doing it at some low cost. Now, I guess everyone here will know that all the mass in biomass is pretty much, in polysaccharides. If you look at a pine board up close, in an electron micrograph, you see these long fibers. These are primarily cellulose, which is just beta-1,4 linked glucose that's hydrogen bonded into these fibrils, about 36 chains per fibril. Additionally, I think I'm running a little bit low on time, but let me just say, additionally, there's about 20 to 30% of the mass is this polyphenolic material, lignin, made from phenylpropanoids. They're polymerized in a free radical process. And as you can see, the presence of lignin is very inhibitory to the enzymatic hydrolysis of biomass, which is showing a negative relationship here pretty strongly. Now, the way these things fit together, nobody really knows, I would say, but this is my favorite. This is how I think it fits together. So, drawn somewhat to scale. The cellulose fibrils are shown here. In this case, they're all pointed in the same direction, which would be true. They're coated with hemicellulose, single polymers of hemicellulose, beta-1,4 linked xylan. And then the xylan is cross-linked to lignin. And the lignin, more or less, we're not sure where it is, but it certainly is more abundant on the outside of the cell than near the center. So, and there are quite a few different linkages holding it together. So what happens, so just to get a sense of it, what the pre-treatment is doing is that the cellulose is completely resistant to the acid, but pentose polysaccharides, the xylans, are actually quite susceptible, and they get fragmented by the hot acid treatment. And that in turn, because they're linked to the lignin, allows the lignin to be essentially washed off the cellulose, and then it becomes available for enzymatic hydrolysis. So as to what we're going to really be able to do about this, I'll show you in a moment what we're trying to do on the chemical engineering side. But there's been a lot of talk over the years about the idea, well, maybe we could, reduce the lignin or modify the lignin, and I participated in that, I have to say. But I think my current opinion, now that I've crossed over and become a chemical engineer, is that it's going to be very challenging anytime soon to actually engineer plants for modified processability. I think long term there is an interesting opportunity to create some modified linkages in the wall, but one of the big problems with really making changes is that we don't have a very good sense of how anything is made in the wall. So for example, when we wrote the genome paper that was talked about in the previous talk, back in 2000, we estimated we could see at least 1,000 genes in the genome of Arabidopsis that look, we're candidates for being involved in making cell walls. And yet, today I think from this 1,000, we would probably only know of a biological function of 24, something like that. So we have a huge uncertainty as to how the wall is made, and it's pretty tough because there are so many polymers, there's so many glycosyl transferases and modifying enzymes that the biochemistry turns out to be very challenging, and that's even true of cellulose, which is relatively simpler than some of the others. So although I think it's a very worthy long-term goal, I think in the short term, we're not going to be able to rely on improving the biomass by direct genetic modification as far as I can tell. So, therefore, most of the emphasis in the field these days is on modifying the process, and one of the key factors in the process is cellulase, of course, shown here trying to extract glucan from cellulose. And although numbers are a bit hard to come by, but in the field right now, in general, people are talking about using up to 50 grams, actually above 50 grams of cellulose per gallon of cellulosic ethanol. That's an astounding amount of enzyme if you think about it. So just do the arithmetic, 65 billion gallons. How much enzyme is that? It's mountains of enzyme. So something fundamental is going to be required to fix this, and I guess Jamie talked to you earlier about at least one strategy to try to find more active cellulases. I think there are fundamental problems here, and one of the most interesting things I bumped into recently is shown here, that in the sequencing of, I've forgotten the organism now. Anyway, recently there have been some small proteins identified in sequencing projects that have quite profound effects on hydrolysis. So for example, in the sequencing of Trichoderma ricei, a protein called swolenin was identified. And swolenin looks like a family of proteins in higher plants called expansins that have been implicated in loosening up cellulose. And I think that was a shot heard around the world in the cellulose field, I would say. Additionally, there are other proteins like this one, I think it's called P21, I wish I could remember the organism, a microorganism. And you can see that when you add it, in this case, to a chitinase, it greatly stimulates the activity. So presumably this thing has some sort of ability to melt, locally at least, chitin. And such proteins are known for cellulose as well. So there are interesting things to be discovered out there. And so, the general approach that I think is we're using, and in collaboration a lot with JGI, is using metagenomics to explore what natural degradation systems are doing. Of course, Phil Hugenholtz here has already published the first termite meta-analysis, and the second one is coming along. Additionally, there's lots of additional sequencing to be done, and the hope is, of course, that in sequencing, cow rumens and forest floors, that we'll spot additional enzymes and then can use the tools of synthetic biology to actually characterize the corresponding proteins. I really like this particular piece of data, which just was published from partially JGI and some collaborators. In this case, characterizing, this slide at least is showing a part of the analysis of the carbohydrate active enzymes from T. ricei, white rot fungus, and brown rot fungus. And And the nice finding, or what I found the exciting finding, is that a whole bunch of very important cellulotic enzymes are missing in the genome of the brown rot fungus. So you can see, for example, there's no CBM1 type of cellulase, and down here the purple ones, I guess those are endoglucanases are missing. Some xylanases are missing, the brown ones. The point being, I think that the authors of this exciting study concluded that the brown rot fungi clearly have a different strategy for hydrolyzing biomass than the white rots and the filamentous like T. reesii. And so I don't know whether this is the tip of the iceberg, but it's a very encouraging example that by going in and doing a deep analysis of organisms that make a living by degrading biomass, we may be able to gain completely novel insights. Of course, this is going to have to be followed up by functional work, but I consider it right now one of the most interesting thrusts in this general area. Now, another one of the big problems in the field is that the organisms that we've developed during the last 5,000 years for making alcohol, well, Saccharomyces at least, only more or less uses glucose. And biomass has a lot of xylose, the most abundant sugar, second abundant sugar. You can see in this analysis from NREL that if you only use the glucose, they were estimating at the time they did this, you could make cellulosic ethanol for $2.40 a gallon, but if you could use 85% of the xylose, you could drop $1 a gallon out of it. That's a big deal, obviously. And in fact, of course, ways of doing this, organisms that do it naturally are known. There's pathways for both glucose and xylose to ethanol. And so what's happening right now, of course, is that there's two general approaches. One approach is take organisms like Pichius stiptis or Zymomonas mobilis that can use all the sugars and try and adapt them to large-scale industrial fermentation. And that they have lots of problems, and so the approaches that are being used, to the best of my knowledge, are whole genome system biology analyses, for example. So at Berkeley, Adam Arkin has made a tagged collection of Zymomonas in which they can do competition experiments with populations where every gene is tagged and under real-world conditions, throwing them into biomass hydrolysate and seeing what each of the genes contributes to survival under those circumstances. And I think that's also a very exciting approach that we're going to need if we're going to adapt new organisms to producing fuels. And the other approach, of course, is to introduce the pathways into Saccharomyces or other organisms. And that's been done. Nancy Ho, in particular, started a long time ago and has had a lot of success. But we're still far away from it. But this shows some data from Nancy Ho's group. But the glucose, in this case the modified Saccharomyces, uses all the glucose in 6 hours. But the xylose, after whatever this 50 hours is still mostly there, actually. And so, although it's a proof of principle, we have quite far to go in terms of solving this. We need to know all the transporters, for example, and we need to balance the redox and possibly other things. So I think this can be done, but it certainly is going to require additional work. Now, I mentioned almost at the beginning that we're approaching the limit for ethanol. Remember I said the corn farmers think they themselves can produce 14 billion or 15 billion gallons of ethanol. In fact, I think this year they're going to produce between 8 and 9 billion, and by 2012 they think they'll be up to the 15 billion limit. So actually, two things can happen. The field will stop at that point, mature, or we're going to start making other fuels. Or I suppose the car companies could declare that, oh no, it's okay, we could actually use 12% or 15%. That's possible as well. But the long-term option, of course, is to make something other than ethanol, and that's probably the most interesting, scientifically interesting option. You can see on this slide from Jay Kiesling and colleagues, you can go glucose to alkanes, or glucose to isoprenoids, or glucose to mid-chain alcohols, all of which are actually quite good fuels. And indeed, there's several companies already pursuing, actually quite a number of companies pursuing these options. So I think that based on the preliminary results, both from the academic world and from the little companies that are pursuing this actually seems quite promising, although I think it's going to require also quite a bit of both new biology as well as quite a bit of synthetic and systems biology to really make it work. because in contrast to everything else we've ever produced from sugars, the efficiency with which you need to produce fuels in order to compete with the existing fuel system requires perfection. So I think it's going to be an interesting challenge going forward. One of the nice things about making these alternative fuels is they go directly into the existing infrastructure, so unlike ethanol, but also they remove energy from the production, because one of the energy sinks in cellulosic ethanol is the dehydration at the final step, and these fuels such as alkanes and isoprenoids don't require, and even butanol, don't require dehydration at the final step because they partition out of the aqueous phase spontaneously. So there's many reasons, and if you ask me what the future looks like, I would say we're clearly going to be making fuels beyond ethanol in the near future. In that respect, my final slide, I just want to say, although I have talked about some of the issues in bioconversion, actually it's entirely possible that there's not going to be any bioconversion. There are ways of going directly from biomass to sugars without enzymes, and from going from sugars to quite good fuels without microorganisms. This is a technology that ultimately ended up in Virent, a company that's commercializing this kind of reforming. I think this is so, even in the EBI, we're running what I would call pure chemistry in parallel with biochemistry or bioprocessing to see which of these will ultimately be most productive. I like this chemistry quite a lot because it's very conservative of carbon. So unlike, for example, conversion of sugars to ethanol where half of the carbon is lost, this conserves carbon, and so it reduces the ecological footprint of producing that carbon. And you end up using biomass as a carrier for hydrogen, which is introduced at several steps. So this is actually, this type of chemistry represents what you might call a fusion of biofuel and some sort of other source of energy. Probably, I'm going to speculate and say probably production of hydrogen from fossil fuel combined with geological sequestration of the CO2. So I think there's a number of options going forward. My final slide is to say that we've kind of we're just getting past the point where this is theoretical. So there's more than 20 companies now scattered around the United States that claim to be either building or already making cellulosic ethanol. You probably can't read any of them, but some of them definitely are. BP made their first gallon about a month ago, and they're already building their second generation facility. As to how long this is really going to take, people keep asking me, when will we make the first 1 billion gallons of cellulosic fuel? I actually think it's quite some, even though there are no barriers to doing it. I think it's going to be 8 years before we see the first 1 billion gallons a year. And the reason is that these facilities are all using different technologies. They're being subsidized in part by the government, by matching grants, and they're built rather small because most of these companies are trying to prove their technology vision. And so they won't build another one until they've got that one running. And on the average, it takes 3 to 4 years to permit and build and proof one of these bioconversion facilities. And then when they've done that, they'll try to build a commercial scale one. Let's say, they'll go from 2.5 million gallons to 100 million. Actually, they would never do that. They'd go from 10 to 100, perhaps. That will take another 3 to 4 years before they get one that's running at commercial scale. So unless something happens, it seems to me we're going to see a couple of generations of these kind of small scale or even commercial scale, but one of a kind, before it actually starts replicating to the 600 plus facilities we really need in order to start making a dent in our fuel needs. So there's lots of time to have impact. I think that's the bottom line on what I think. People keep asking me, is it too late to actually change this? And I think the answer is definitely not. There's an opportunity to have a lot of impact right now. All right, I'd like to stop, and I'd be happy to take any questions. Thank you very much.
Have questions for Dr. Sullivan? I guess I'll start with one. Given the uncertainties in engineering of large-scale cellulosic biofuels plants, one can imagine that these companies are risking substantial capital, and if you guess wrong, or new developments come along that make the plant a better plant than the one that you build, what are their options, and what's the strategy for getting around that? Because you do need to get out of the blocks to learn how to do this.
You know, that's a great question, and I think many people are asking themselves that. So, you know, I think that what might happen is that fuel is the cheapest stuff you can make. It's just there's a lot of other valuable materials of large-scale industrial use that could also be made from inexpensive feedstocks. So I would be surprised if many of the companies that are building these don't have a second strategy, which is, you know, if we can't make fuel at cost, maybe we could make this other industrial synthon at cost, and maybe 10 million gallons a year would be just right for that market. So I think there are a lot of opportunities to do that, and we might see that the first generation facilities end up getting diverted to specialty chemistry from biomass. And then still the teaching or the learning will take place with these small scale facilities, and some of them will succeed and will be scaled up, but probably of the 20 odd technologies that are being used right now, there will be one or two winners, I would think, in the end. Of course, there's deep capital coming in. So I was at a meeting this week where we had BP, Exxon, Shell, and Chevron, and Danisco, and Novozyme, all in the room together, learning about biofuels. And so when you see Exxon in the room, stand back. You know, it's like something happened.
Yeah, Dr. Somerville, you mentioned the inhibitor products that are going with dilute acid pretreatment.
Yes.
What are the workarounds that you see for that? And I was wondering where alkaline pretreatment falls.
Yes, well, alkaline pretreatment, Bruce Dale is trying to scale up what's called an AFEX process, and it's an interesting, it's a pressurized ammonia, and so what he does is he infiltrates biomass with ammonia under high pressure and temperature, and then explosively decompresses it, although he doesn't like to use the word explosive. That actually doesn't cause inhibitors, and in fact you can feed that material to cattle. It's so non-toxic, and in fact there's some interest in creating some variant of that, because you could strongly increase the feed efficiency of a lot of biomass if that could be implemented. We don't know whether it can be done, because the explosively decompressing 200,000 liters of ammonia is scary, because you can't just off-gas it into a big hood in the sky. You pretty much have got to capture it and reuse it, and the engineering for that looks really challenging. The approach we're taking is to try and identify better catalysts. So instead of just using sulfuric acid, we think that there may be some structured catalysts that are capable of hydrolyzing glycosides, but don't dehydrate sugars, for example. In terms of the acetic acid, we would be open to suggestion as to how to get rid of acetic acid. We're also using ionic liquids. So we have a group of chemists that are both using testing known ionic liquids, and as well as developing new ones that could be recycled on the scale to which we would need to do it. So ionic liquids will decrystallize materials so that you can actually skip the acid hydrolysis completely. But the challenge there is to be able to, you have to have essentially 100% recovery of the ionic liquid to make that work.
Yeah, I just wanted to add, this is 2009, I think, DNAF. We solved the explosive problem by changing the balance, ammonia expansion.
Yeah, right.
Yes.
Right.
Hi. It seems like you're right with Professor from Sapphire about the difference in yield of biofuel crops versus cellulosic fuel crops. Cited the number of like 5,000 gallons per acre algae in
Yes. Algae can have very large numbers in a flask, and so I think the challenge is to see what they can do out in a stirred pond. Maybe Sapphire's already done that, I don't know. If they did, are they claiming they're getting that yield in practice or not? I couldn't quite hear you, I can't quite hear you, I don't think...
I was just curious what your